Okay, so we have been speaking a little bit about the interactions that our R groups can create. So those electrostatic interactions, that hydrogen bonding, those dipole to dipole interactions, even the disulfide bridges, the ion bonds that can form, uh, all of those help create a protein that's got very specific properties. Now, the areas where we've got very low or no overall charge, or even dipole to dipole interactions, those areas of low charge are very hydrophobic, and the areas with high charge are hydrophilic. What this essentially means is the areas that don't have any charge will not want to interact with water. And the reason for that is water is quite an interesting molecule, and it will create hydrogen bonds. And we spoke a little bit in the very first chemistry for biology lesson that the water molecules will interact to exclude anything that is non-polar. So what we've got is the hydrophobic regions will try and locate themselves and hydrophilic So, water is polar, and it makes up the vast majority of the entire cell. And it's got a negative charge, a slight negative charge on the oxygen, and a slight positive charge on both hydrogens. Now, what we haven't spoken about for quite a long time is cell biology. Now, if you are in S1, you would kind of have this image of the cell. And as you have progressed through school, we've kind of added on bits and pieces to this. We're going to add a fair few more bits to the cell structure in advanced higher, but we'll be getting onto that slightly later in the course. However, if you've got this image in your head, we can certainly work with this. So this water molecule makes up most of what you would have called the cytoplasm before, and very specifically we call this the cystole. The reason why we're going to split the cytoplasm up into two different things, the cytoplasm really is the cystole, and the lumen of the internally bound membrane structures, which we'll get onto later, but I wouldn't worry too much about that. Where we've got our hydrophilic section really in the cell is this entire area. So everything that's hydrophilic is kind of the cystole. Now that means that any proteins that are hydrophilic will locate themselves within the cystole. However, the hydrophobic sections are maybe a little bit more interesting, and proteins that are built up of amino acids that are mainly non-polar will be excluded into an area where there's very little water molecules and most of the surroundings are non-polar. So what we've got to do there is we've got to look at our cell membrane. Now, our cell membrane is made up of phospholipids. So this is our, and by this time we're kind of on to National 5 we've got this image here in our heads. Now it's a bilayer, which means I'm going to have to do another layer down here. And you may well remember, hopefully, that there's a head section that is hydrophilic. So that is the head and that is the tail. And the head is polar and the tail is non-polar. So any sections of a protein that are hydrophobic are actually going to want to embed themselves in the tail region of the membrane. So they're going to want to be somewhere within here. Now, when we come on to look at that, that's going to be really important for anchoring some proteins on the cell surface, because the cell surface is really an area that's really interesting. It's how the cell's going to detect its external environment. It's going to be giving the cell a huge amount of information about what the cell actually needs to do to help the body function correctly. So those cell membrane proteins that anchor themselves in there, the section that's going to be in the bilayer actually sandwiched between the polar head sections, that's going to be a non-polar hydrophobic section of the protein, and we'll talk a little bit about how that comes about. First of all, what I want to do though is show you the actual phospholipid. We're going to dip into a little bit of additional detail here, but I think it'll help you see what's going on. Okay, so our phospholipid is actually an interesting structure, and it's not ubiquitous, which means they are not actually all the same. What I'm going to do is I'm going to give you an example of one type of phospholipid that a cell can create, and we're going to talk about how that structure is important for creating that bilayer. So our phospholipid is certainly going to have a phosphate somewhere. That lipid really means fat. So it's going to be like a fat molecule with a phosphate on it. So we have an atom of nitrogen that's got a positive charge there. And like I said, not all phospholipids are exactly the same. And what you'll find is that depending on what type of cell it is, it may express a different membrane. So it may have a different ability to carry current, and that'll be really important for things like nerve cells. So not all of our membrane phospholipids are exactly the same. That actually is, to me, a really interesting little, like the skin of different cells is actually really quite different, which to me is quite cool, and I think there would be lots and lots of interesting little bits and pieces of information about that and why it's important for different cells to have different phospholipid build-up. So this is actually our phosphate that I'm doing here, and what you see up here, this whole bit's going to be quite polar because we've got a big positive charge on that nitrogen, and here we've got a negatively charged oxygen, and I've got oxygen down there, and carbon, a hydrogen, then we go this way. I'm not doing this from memory, by the way. I do have a picture of it in front of me, but I thought it would be helpful to actually build it up for you, so you could see it. So this is the bit that's interesting. This is actually, well, the whole thing's interesting, but this bit is an ester bond. So if you did higher chemistry, you'll have done ester bonds, and this is how we attach our fatty acid chain. Now it's that fatty acid chain down here that I've kind of run out of space for a little bit. This bit is non-polar. So this is all, in fact, what I'll do is, this is all carbons and hydrogens. And these tend to be very non-polar, and they stretch down, creating the tails. Realize this is a horrendous diagram for your imagination currently, but hopefully these bits can yank onto there. These are all non-polar. So when we took a look at our different amino acids, there were some amino acids with these aliphatic hydrocarbon R groups, and those tend to be non-polar. So if you've got a big block of glycine, certainly one, alanine would be another, isoleucine would be quite non-polar, leucine would be quite non-polar. So if you are looking at those amino acids, you're looking at sections that are going to be pretty non-polar, and they're going to want to embed themselves in that phospholipid bilayer. And it is interesting to note that not all the phospholipids that you find, so not all of these things in the bilayer, not all of these are the same. So, and it'll be usually the head will have some other thing added onto it or taken away from it. It might have different atoms that make up the head section. So this section here, really that bit, is the head section, and this bit here, right the way down here, and these go quite far down. That's our tail section. So that's our phospholipid, and that's how we draw it at National 5. Even at Advanced Higher, you don't need to have that picture in your head. What I would probably have in my head, just to help me, is positive and negatively charged head section. That's where my phosphate is, and what I would have in my head is just a aliphatic or hydrocarbon tail section that keeps going down, and that's non-polar. That's as much as you need to know at Advanced Higher, and to keep it in your head. It is about taking lots of information that's complex and making it a bit more bite-sized and manageable for yourself, especially at this stage. There's lots and lots being thrown at you, and it will take us a whole year to build up our understanding and our knowledge enough to sit that exam. And the exam will not be, if we go through this properly and we do it right, the exam should be pretty straightforward. We'll speak about some techniques closer to the time, but the stuff that I'm giving you and the way that I'm giving it to you is pretty full on, and it's pretty as much as you would be expected to know on the absolute top end of your understanding and your knowledge. I'm not leaving much out, and it's probably a little bit of overkill, some of it, especially some of the PyMol stuff. We could have gotten away with not doing it, but I think it is good to do it, and it's good for you to have a little bit of an understanding of what those phospholipids are, and that they are not all the same. In between cells, or even within the same cell, there can be pockets that this head section is made up of slightly different atoms, and it will have a slightly different charge, it'll have slightly different features, it'll have a slightly different effect. Right, so on to the next bit. Okay, so globular proteins. I always found this a little bit weird. We've got lots and lots of different ways of talking about proteins, and really this is a way of talking about almost a half of the types of proteins we've got. We've got globular proteins and we've got fibrous proteins, and we've got a whole spate of things that are kind of in between. But the globular proteins really just talk about proteins that are either squished into a sphere, or a little bit of a sausage shape. Almost all the proteins that we talk about are globular. Enzymes are globular. Transport proteins could be argued to be pretty globular. They're a little bit on this end of things, but they're quite globular. And they're all kind of quite, they can have lots and lots of amino acids, but they're actually quite restricted in their general length. So they're not going to stretch the expanse of a cell. However, in fibrous proteins, you really can get these very, very thin, extremely long proteins, actin and myosin in muscle cells, which I'll put a link into some of that stuff, so you can see it, but we don't talk about them a huge amount. It's really the globular proteins that we're focused on. Now, their shape, a huge part of the reason that they form the shape that they do, and we've covered this hopefully to the point where you can kind of see what's happening. On the outside of the protein, we're going to have water molecules, and those water molecules are polar. So the hydrophilic sections, so the sections of the protein that have a charge, will face outwards towards those water molecules, and the hydrophobic non-polar sections will all cluster in. So if the tips of my fingers are non-polar, they'll kind of hide away from the water, and the backs of my hands and the backs of my fingers, those sections will be polar and they will face out towards the water, and you'll get this kind of globular shape. And that's all very important in that tertiary structure when we produce the polypeptides. These are both globular. This one's a bit more sausage shape. To put it in perspective, a globular protein would maybe be like this kind of cluster, whereas your fibrous proteins are going to be absolutely huge, and they're going to stretch. They're very, very thin, and I've probably not done a good job showing you quite how fat that globular protein can be compared to the fibrous protein, but these proteins can be extremely long, and the globular protein really could contain just as many amino acids, but they're all confined into a smaller area. So that is our globular proteins, and when we say globular proteins, we're really just talking about a huge group of proteins, like our enzymes and our transport proteins. Lots and lots of proteins that perform very specific functions throughout the body are globular. The fibrous proteins will be found in things like muscle fiber. So once again, this concept of polar and non-polar comes in to creating our structure. So what we're going to go on to next is really seeing how these globular proteins, with their polar sections and their non-polar sections, interact at the cell membrane to give us cell membrane proteins, and how they anchor themselves into our cell membranes. And this will start to hopefully build up the picture of what our cell membrane really does, because it's not just, as much as we've got that image of a cell, and my membrane's gone a bit weird there. In fact, we'll make this a protein. It's got a wee protein here, a wee protein there. As much as we've got this image of the cell, and a lot of the time we can think that most of the action is between the nucleus and those ribosomes, a huge amount's happening on that cell membrane surface, and it's really interesting. It's really the gateway to the environment that our cell has, and it's got a nose and eyes and the ability to feel in some respects what that environment is telling it. So it's a really interesting area of study. I've actually got a paper that is much more readable than the last paper that I put out, and that'll hopefully take you through the history of the membrane. I've only read through some of it. I'm actually finding it really interesting. I'm reading it as well, but I've only made it a couple of pages in. But it's really good, and if you get a chance, once again, the papers are certainly over and above. They're really me trying to slowly introduce you to papers. And the reason for that is when I got to uni, and I had never seen a scientific paper at school, and I'd never been introduced to them. And we got our first essay, and I remember reading it and just being like, I am lost. I don't know what half these words mean. I feel like I don't have a clue. I felt a bit overwhelmed by it, and what I didn't realise was diving into the method was not required. You're not going to know a lot of the words and the nomenclature they're using, and part of reading it is learning that stuff. And I think for me, it would have been nice to have had that pointed out instead of this feeling of you should just be able to grasp this off the bat, which I wasn't able to do. I sat staring at the paper being like, this seems way above my head, which, I mean, you are going to find yourself out of your depth, and it's about learning how to swim, I guess, a little bit with it, and how to cope with the fact that you're not an expert in a lot of these things, and becoming good at these things does take time, it does take effort, and you just got to keep hacking away at it. I still don't know anything really in the grand scheme of things. Whatever you do know is always going to be a sliver of what there is to know, and it is about coming to terms with that, because I do feel like at school we very much teach in a way that it makes the appearance like we know everything that's going on, whereas really we've just got a general overview, and the details are pretty misty for most of us, and you need to constantly be reading, need to constantly be trying to understand it, and that's all it is. And if you're constantly trying to do it, you will be doing the right thing. So, right, before I go on a massive ramble, we'll move on to the next bit. Okay, this is definitely one of those bits of nomenclature that can get a little bit tricky, because biochemistry is a little bit tricky, and I've got to be quite careful how I talk you through this bit. So membrane proteins, we can branch into two major subdivisions, peripheral and integral. The peripheral proteins interact pretty weakly, and they interact in a temporary manner with the membrane. Remember, the definition of that membrane is not just the phospholipids, but it's the proteins plus the phospholipids that make up the membrane. So those peripheral proteins can either interact weakly with the phospholipid bilayer or some of the proteins on the surface itself. They will only ever penetrate one side of the bilayer, and they can leave, and part of their job might be to leave the membrane itself. The other ones are integral. Now, these should not leave the membrane at all, really. They will, because your cell, these proteins have a life expectancy, a sell-by date, so they will need to be removed, broken down, and replaced by new ones. However, their job is not to leave the membrane. Their job is to stay in the membrane and do a function. These integral proteins can either span the entire depth of the membrane, so I'll show you some diagrams of that in the next bit. However, those ones that span the entire way across, so they go from one side of the membrane right through to the other, those are transmembrane proteins. Not all of the integral proteins are transmembrane. Like these ones, some of them will only penetrate the bilayer on one side. However, in the case of the peripheral ones, they are designed to leave the membrane itself. The integral ones, if they're not transmembrane and they're still considered integral, they won't be designed to actually leave the membrane. So I'm going to show you a couple of different examples of these. The Wikipedia page for this is actually really quite good. It's got a really nice diagram on it if you want to read further about this. But I'll give you the little diagram and hopefully that should allow you to see what's going on here. Okay, so hopefully this clears up any potential confusion, as I appreciate there may be potential confusion with something that's a little bit going a little bit deeper than we've gone before. So these two are both integral proteins. That means they're not designed really to leave that membrane. This integral protein here, this bit is going to be polar and can interact with the extracellular matrix, basically the watery substance on the outside of the cell. The hydrophobic section is going to come in and embed between the bilayers. Now, what I've done in this diagram is I've shown you that the anchor point between the bilayer is quite large. So that's going to help stabilize it and force it to stay in this position. This is a peripheral protein that's interacting with the bilayer, and it is embedded in the bilayer, but it doesn't go very far down, and the hydrophobic section is not very large, so that might allow it to leave, and that's why it's considered peripheral. And it's not as well embedded as this one. They are both still embedded, but this one is designed to leave, this one is not. Now, the other type of integral protein which goes straight through the bilayer, that's a transmembrane protein. So this protein, this entire section is, well, essentially the entire section from here to here is non-polar. And that is hydrophobic. It'll want to sit within that bilayer. And these bits here will be polar. And this is the cystole, so the inside of the cell itself. So those are your definitions and how to view those proteins there. Now, what I was going to do was go on and talk about ligands, but I think, honestly, now that we've done the transmembrane proteins and we've spoken about them, I've taken a look at the ligand section, and it's probably a lesson in and of itself. And there's some really interesting bits that we can talk about in terms of the interaction with DNA. So I think what I'm going to do is probably put that together over the weekend and do another quite big video using PyMol as well. I think I'd like to try and integrate some of that into it. Obviously, with the videos that have slightly more editing and slightly just better, higher quality stuff, they do take a little longer than these ones. So this one is a straightforward one just for notes. We will hopefully we will get Q&As started from today. I'd like the reason that I'd like to do the Q&A sessions is that I am actually really missing the feedback that I get from classes in terms of how people are picking it up, how people are discussing it, what questions are being asked. I'm just kind of trying to put it in, put the information out, I should say, as straightforward a manner as I can, but I'm not able to to deal with any of the intricacies that we usually do when we're learning. So I'm looking forward to the Q&A, and hopefully any issues or anything that we want to ask from the videos, we can do that going forward in the Q&A sessions, and might be able to even record some of the really good bits and put them together as a lesson. But this is it for today. So today was just the phospholipid bilayer, the globular proteins, how they interact in a hydrophobic and hydrophilic manner, the membrane proteins, the peripheral and the integral, and remembering that there's really two types of integral proteins. Some are transmembrane, some are not. Those monotopic and polytopic are there. You don't need to know those. That's why they're in quotation marks. But some of the nomenclature refers to them like that, so it might be helpful if you're doing outside reading. I'm going to put up with this lesson, I'm going to upload a paper that takes you, actually, that was the one I was speaking about at the beginning. It takes you through the history of the discovery of the cell membrane and cell structure and cell biology itself, and it's a really good paper. Once again, we're dipping our toes into the scientific literature. The language is something that we are learning, and do not be hard on yourself. It might take you a while to read through it, might take you a while to Google words. That's all part of it. Hope you guys are doing well, and I will hopefully see some of you in the Q&A.